Hello and welcome to Feedstuffs 365. I'm Sarah Muirhead. One of the major problems with assessing African swine fever virus contamination and the risk of transmission in the global feed supply chain is the lack of a system to accurately measure, monitor, prevent, and control the virus. Joining us today to discuss various analytical methods for ASFV and to talk about a newly developed surrogate virus assay for ASFV are Dr. Jerry Shurson and Dr. Declan Schroeder of the University of Minnesota. Welcome to you both. Great to have you with us today.
Well, thank you, Sarah. It's great to be back.
Yeah, thank you, Sarah. Thanks for having me.
As we get started, a reminder to those of you that are joining us, put any questions or comments that you have there in your chat box off to the right of your screen. If you're joining us from social media, Facebook, Twitter, LinkedIn, be sure to put your questions or comments there and they will come into our studio as well. You'll also find posted already in the chat a related article, as well as a link to a webinar series that we recently did with the University of Minnesota, where this topic was first covered. So Jerry, you've been on a few of our Feedstuffs 365 sessions before, and you've been sharing some of your team's work when it comes to assessing the risk of contamination and potential transmission of African swine fever virus in the global supply chain. Provide for us an overview, if you would, of some of the challenges that we need to overcome for controlling the potential of ASFV transmission in feed.
Well, thank you, Sarah. And I think one of the things that Declan and I and the rest of our team has really learned over the last several years, where we've really been working hard to try to better understand this very unique virus, is that there are three major themes that come into play. The first one is we're dealing with a lot of uncertainty. Uncertainty about whether or not feed ingredients and feed supply chains, especially imported ingredients, are even contaminated. And a big reason for this uncertainty is that it's really difficult to get accurate measures of virus infectivity. This is a very unique virus. It's a megavirus, and Declan's going to tell our story about a little bit of what we've learned here in recent years. But to go along with this uncertainty of whether or not feed ingredients are contaminated, we don't have accurate measures that allow us to determine the concentration, the survival, the infectivity, or even the prevalence of potential contamination in various feed supply chains. And so I like to use the term, to go along with uncertainty, the term measures matter. And I think you'll find that to be the overarching theme of what we're going to be talking about during this webinar today. And the third theme is that things aren't always as they seem. One of the big surprises, I guess, and Declan's going to explain this in greater detail, but just to get everybody tuned in, we've always known that African swine fever virus is pretty resilient or resistant to time and temperature treatments as mitigation strategies. What we didn't realize is how much time and temperature it really does take to inactivate this virus, and we're going to share some of our recent findings with the group today. And I think what this really means to the feed industry and to all of us that are trying to develop improved biosecurity strategies and mitigation strategies is that maybe our previous assumptions aren't quite as valid today as what we think they are. And I guess really what we're going to be talking about is sharing some data causing us to rethink some of these strategies as far as time and temperature mitigation and what we've learned so far. I think it's also important to realize, and many of our audience members today probably are aware of this, that last year the FDA came out with some new guidance for the feed industry, and for the first time ever indicated that viruses now are categorized or classified as reasonably foreseeable hazards in feed ingredients. That That's the first time this has ever been brought to bear. And so I think we're all really pleased about this, because I think we recognize that viruses can be hazards or potential contaminants in feed ingredients. Certainly African swine fever virus is one of those. But again, if you look at the bottom of this figure in the lower right-hand corner, FDA classified viruses as relatively low resistance to heat compared to bacterial spores or some of these other categories of biological contaminants, which in the case of African swine fever virus, we know is totally contrary to what's known about the resilience of heat. And so, here's an example of things aren't always as they seem, so to speak. And certainly when we get to thinking about the need for implementing a preventive control plan for African swine fever virus and feed supply chains, again, there's still some uncertainty. It's great that this topic has been brought to the forefront. And I think we all recognize that in terms of severity, this type of event would be ranked as medium or critical in this decision-making matrix. But I think one of the uncertainties is, is it a low or medium probability? If it's medium probability, the probability will occur in time if it's not corrected, or if it's a low probability, it may occur in time if not corrected, or possible to occur in time if it's not corrected. Because of the uncertainty and the lack of ability to measure the prevalence and concentrations and infectivity of this virus, it makes it difficult for a feed mill manager or owner to know, do I really need a preventive control plan or don't I? And I would caution everyone and recommend, of course, that we do put one in place using the best biosecurity methods that we know of today. But even in doing so, in actual compliance with a preventive control plan, it implies that we have an ability to monitor and take corrective actions if a contamination event occurs. And so again, this ties back to my second theme of measures matter. We don't have a method or analytical procedures that allow us to do this at this point. So we've got a lot of challenges to overcome. And our group has been working really hard over the last few years to develop a new strategy, surrogate virus strategy, and help us realize that the measures that we use and the interpretation of the results are really critical for the kinds of mitigation strategies that we choose to implement in our feed supply chains.
Declan, as Jerry briefly mentioned, measuring African swine fever virus contamination in feed and the accurate interpretation of those results, it's not easy. It can be very confusing. Help us understand the difference between the common analytical methods and the measures being used and their limitations.
Right. So, African swine fever virus is not present in the US, right? Thank goodness. And as a result, when one tries to understand and work with this virus, you have to work in a restricted environment. And so in the US, we have, it's a select agent status, and so we work with this virus in the BSL-3 program. As a result, your tools are limited. You can't just do the experiments you want to run. So if you're trying to ask the question, is feed a problem? Can feed cause disease? Well, then you have to think about an animal model, right? You have to have a virus in the presence of feed, and then you feed that to an animal. And then the animal can be either in a wild setting where it exists normally, or it's in a laboratory setting. And those two experiments are really, really challenging in the US. We just can't, we have really limited space and access. So you can't do the animal in the field, first of all, ASF. It's just impossible. So here on the slide on the right-hand side, we talk about in situ, so it's in the environment. Animals are naturally infected. You just can't do that experiment, especially here in the US. Whereas, of course, in a country that has ASF, you can certainly under controlled environment do that. But even then, if you can, it's difficult because it's a size issue, right? You can't work on thousands of animals, and all animals are different, just like every human is different. We have those who are more susceptible, those are less. So it's a really complicated dynamic, right? And then you bring them into the lab, so it's like an in vivo study. It's not a natural situation. The animal will be fed something under very much more controlled environment. But again, due to lack of space, you can only work with 1 or 2 animals. And so you don't get the feeling of the size in terms of population. Then you then say, well, actually, let's try and really control this and scale and get replication in. And so then you reduce the complexity of an animal and take it down to a cell culture. And so we have a variety of cell cultures that we can use to do this infectivity. What's common to all this is that we're actually just measuring an infection. We're not measuring the virus. And a lot of the studies work on ASF really is restricted to the fact of, can you see an infection or can't you? It doesn't mean that you can actually say the virus is gone. It doesn't mean that the virus is doing what you think it's doing, because you're not monitoring the virus. And so, the layers of complexity in terms of how one runs these experiments also should be seen in sort of the different molecular tools one uses. And so in the slide, you can see these various green circles where I've highlighted the sort of methods we use to try and measure infection. In the end, all these assays all work together with an animal study, with a cell culture study, that only speaks to an infection. It doesn't speak to how the virus is actually behaving. So this is one of the big limitations in my mind that we needed to overcome. And secondly, try to work on a system that is beyond just ASF. You need to be able to work on a system that you can work more freely and do larger scale studies.
I recall from the University of Minnesota African swine fever webinar that we did a few weeks ago together, you briefly mentioned some exciting new discoveries that have led to the development of this surrogate virus assay for African swine fever virus. And you're calling it, I believe, RISNA. I believe you noted that you're using a viability PCR analysis for more improved assessment of the characteristics of these megaviruses. Explain this new assay and analytical method, and why are they an improvement compared to the others that you just described?
Right. So if you look at this slide, for example, you see I've shaded the polymerase chain reaction, PCR, people use a lot, right, as a method to detect the virus. So this is where you don't necessarily look at the infection in the context of infection, but you're actually just looking for the presence of the virus. The problem with that reaction is that you only detect the genetics of the virus. So this is a DNA virus, so you only detect the DNA of the virus. You have no idea whether that virus is actually a virus, or whether this is the DNA that's been damaged and now it's sitting, for example, in the feed. So there's no way to assess the threat. So one of the first things we decided to do was say, right, can you improve this assay? And so there's an assay that's been first developed for bacteria, and now is being used more and more for viruses. So there's a few examples of it being used, for example, in PDV. There's for humans, we've used it for influenza, we've used it for SARS-CoV-2. So there are more and more applications. We think we're the first to have developed use this method, Viability PCR, for ASF. And the principle is that, as I said before, the PCR would usually detect whether a virus is whole. So here, imagine this green circles as being a complete virus, functional virus, viable virus that had its DNA intact and is still looking very healthy, as opposed to a virus that's damaged, and so its structures is compromised, or you can have a situation where the DNA is just lying free. That a PCR can't distinguish between what's viable and what's not viable. But what we do in the viability PCR is we add a dye which binds to anything that's free. So here you have DNA that's available because the virus is damaged. So the dye will bind to the DNA. And then you remove it from the sample. And this is done through a light inactivation step. And then the PCR only looks at the viability state. So that's the one dynamic we've worked on that allows us to go beyond just PCR, understand viability, where the particle lies. The other thing we wanted to develop was, as you said, this RISNA assay. So RISNA stands for risk-free because we're working on an algal virus. So in virology terms, viruses are very specialized in terms of how they interact with their host. So a human virus will not infect an animal virus, right? An animal system. Or there are, of course, exceptions where you get viruses from animals jumping to humans and back and forth, but largely speaking, they are separated. Similarly, a plant virus won't infect us. And so over time, and as viruses evolved, we have an algal virus that actually has a common ancestor to ASF. And we can then use that. In this case, we discovered an algal virus that I've been working on for 20 years, and it shares lots of properties. Before I go into explaining a little bit more about this algal virus itself, what I wanted to touch on in terms of the question you were asking about the RISNA, well, what it does is allows us to capture the best parts of all these different systems. Working on a system that's natural. The algae we work on is the animal. You can imagine that is the pig, in a sense, that's being infected. We're not modifying it like in the cell culture. You have to change anything. And this algae behaves like it behaves in the environment. It behaves in the lab as it does in the environment. So you're getting the best of all three types of measurements in one place. And this is the power of the surrogate, and especially an algal surrogate. And so when we work on this virus in the algal setting, we are essentially working on billions of algae in one tube where we can run an infection cycle. And then we bring in these new tools like a lysis assay, a bioassay, where we can actually monitor how these algae are behaving, just like you do in a normal animal study. And then we add this extra layer of excitement so we can actually really work on viability now, right? Now we can actually ask, what's the virus doing in this context? This really changes the ballgame. The combination of this surrogate and this lysis assay allows us to address questions that we cannot do with ASF in a really rapid way and in a safe way, for that matter. And so this is the beauty of this RISNA assay, is that it has this algal virus. And what this virus, if you look at it, ASF has this capsid with this outer membrane, and then it has an inner membrane. For some reason, sorry, the slide has changed, but here you have a situation where the algal virus is a similar thing. There's the capsid, as you can see, it has the inner membrane, and also has this outer membrane which surrounds it. Just for some reason, it's been deleted from the slide. So actually, they share common ancestry, and they really look the same. And then taken together, when we looked at our algal assay, which is actually RISNA, and you compare that to ASF, we actually can tick all these boxes when it comes to understanding and accepting this is a good surrogate for ASF. So, as I said, there's an algal virus, so it's non-pathogenic. There's very similar characteristics. It's the same structure. We can grow it really, really well. We can grow a lot of this virus, and we can grow it safely. We can do large-scale populations because there's millions to tens of millions of algae in one experiment. And it's a genetic stable line. I can go on and on and on about this, but together, this assay really works well as an assay that allows to move beyond what we can do with ASF.
You have an article coming out on this, I believe, and we've shared that there in our chat if anyone wants to go ahead and download that. Now, it's interesting. The title of that article is "Unexpected Thermal Stability of Two Enveloped Megaviruses." ... What do you mean by unexpected thermal stability, and what are the implications for using time and temperature to inactivate African swine fever virus in feed and other substances and materials?
Yeah, right. So, this is where, when you start looking at mechanism, you start to realize we are missing a lot. So, this is what Jerry always talks about. Things are not always what they seem. Mm-hmm. And so here's an example where we thought we knew what we were talking about, and then we realized, hang on, we just don't know what we're talking about. And this is actually the last point here. When you look at a surrogate, one of the first things we wanted to do was to ask whether the virus is susceptible to, for example, temperature, and whether the algal virus behaves just like ASF. And when we did our cell culture method, which is similar to the cell culture TCID50 method, or they had assay method that is used for ASF, we too found the same thing. We found that this algal virus is susceptible to temperature, and anything around about the 50 degree, 60 degree mark will make this virus inactive. And that's because the cell cultures survive, the cells grow, and that was the same for this. But we wanted to then add the viability part to this. And then this is where everything was just gone. The rule book was thrown out. Essentially, what we thought we knew just didn't work. I mean, let me walk you through this is the real focus of the paper, is where we expose this virus, and on the left-hand side is EHV, which is the algal virus, to a range of temperatures 60, 80, and 100. And just as a reference, when you take this virus and you take it from our fridge at 4 degrees, we can do a standard PCR, which tells us, yes, there's 1 million viruses present. So this is log numbers. So the 6 means there's 1 million viruses in our test. And when you run the viability, so where we see V, that's viability. You say, right, it's living at 4 degrees, it's all fine. All the viruses are viable. Now, I didn't put it in this figure, but trust me when I say all these tests where you do 60, 80, or 100, the standard PCR gives you high virus count, because you're just looking at the DNA. But for the purpose of this study, we just wanted to show you the viability. So these are virus particles that are intact. What was surprised to us is that this 50, 60 degree mark, there was still lots and lots of viruses present. There was less than 1 log reduction. We went from 1 million to about 500,000 of viruses still present. So this is structurally intact viruses. So we were then thinking, hold on here, is this misleading? We're not getting death from measuring using the cell culture method, the bioassays. What's going on? What's wrong with these viruses? Or are they functional? Could they work? And what's even more interesting was that if you increase the temperature to 80 degrees Celsius, it didn't change. You only had to go down to, you start seeing some significant changes when you go down to 100 degrees, or we go up to 100 degrees, you'll see 3 log reduction. And so we thought, hang on, what is going on here? Is the cell cultures misleading? And then you also wanted to make sure that this was also true for ASF, and true, it was also for ASF. You can see on the right-hand side, ASF does the similar thing. ASF is also stable at 60. It's also stable at 80. You have to go up to 100 degrees. You have to boil the sample before you start getting this sort of loss of virus number. And lots of people are saying, well, actually, and we're thinking, is this true? Can we trust the viability PCR? Maybe it's a new method. Maybe there's a problem with that method. And then we said, well, let's try another method. And this method is where we take this virus particle and we stain it. And we stain it with a fluorescent dye, which fluoresces green under a microscope. So here on the left-hand side is what this virus looks like when it's stained. And these green dots are all virus particles. Okay, and if they aggregate, they'll produce these strings. But we don't worry about that. What we're essentially saying is there's lots of virus particles. This method only works if the virus is intact. It has to produce a virus particle. Otherwise, the DNA will dissolve and you won't see the signal. When you increase the temperatures, and in this case, here's 100 degrees, you go up to boiling this particle, the sample for 20 minutes, you still see virus particles. There's less than when we started, just like you see for the viability PCR, there's a reduction, but there's still virus particles around. And because this virus has lipids, you can start seeing this red color, which stains for lipids in the background. So we have 2 methods, the viability PCR and this new method of confocal microscopy, which is more time consuming, but certainly the viability PCR is the best way forward for large scale screening. That tells us that this virus is stable at these higher temperatures, something we didn't appreciate before.
Interesting. There's been a lot of interest and a lot of focus on imported soybean meal as the potential concern for ASF virus contamination. And I understand your team has got some funding from the United Soybean Board and other feed industry partners to not only develop this RISNA assay and diagnostics, but also to begin to understand whether these heat treatments that you're talking about that are used in various stages of the soy processing process, if they affect the inactivation of the surrogate virus. Share some insights that you might have on what you've learned so far in this area.
Right. So, temperature is a good proxy for stability. Right. So, in terms of just as a virologist, this is a common method. So, these sort of methods and design was really to understand the sort of structure and structural integrity of this virus at high temperatures. It tells you it's really strong. But of course, temperature in the real world gets used as a way of preparing material. And in the feed industry, this is what we do, right? We try and prepare our feed. And through that process, we are comfortable in saying, right, viruses would be destroyed through the system. But we had no real way of testing this for ASF. You can't take ASF and take it to a plant somewhere, in Minnesota. It's just, there's a non-starter. So how do you go about doing it? So it goes back to the RISNA assay and actually having a surrogate. So the surrogate allows us to do more than just just working in the lab. We can go to the field. So absolutely, the United Soybean Board has been absolutely fantastic. They saw the potential of this assay, and through their funding and through our engagement with other private enterprise within the US, so I've listed their names here. And more recently, we had support from the Fats and Proteins Research Foundation. They've also understood that the potential of this assay. We were starting to design these experiments in a real world scenario. And here's an example where we've taken this virus and we've added to feed. And then we asked the question, how does this viral virus behave in feed? And then we can also take this virus and we can add it to the raw material and take it to a feed mill and run through a process of the feed mill. And together with all this, we can put together kinetic data, infection stability data, and really come and address the question is, how does this virus behave in the real world with feed? And again, this slide just highlights the fact that when one works with the end product like soybean meal, one has to remember how the soybean gets made. And one of the questions we had was that whether you use the conventional solvent extraction method or a organic extrusion type method, there are different steps that one has to go through to produce soybean meal, which requires heat for inactivation. And one of the interesting outcomes, and this is a publication we're currently working on, is that we know through this process that the virus can still survive right up to the hull section. So the temperature treatment at this early stage, the hulls of the soybean can still have virus present, and at various stages we can still see some level of virus. It's still a work in progress. We're still trying to understand the significance of detecting this virus through this process. But it is really a powerful tool. We can go into the system and we can really measure data in real world time, right? And really understand, is there a threat? So not only is it the end product that could be a source of contamination, but the raw product could be contaminated. And then how does the virus survive through this process?
And now you've been using the new RISNA assay for evaluating the potential of African swine fever virus survival in simulated feed transportation models, as well as determining the effectiveness of various commercially available chemical mitigants in feed. Share with us what you've learned in those studies.
Right. So this is an example of us understanding soybean meal through a pilot facility. We then said, well, could you use it and ask the question of, right, we know through a lot of the good studies done by Scott D and his group in Pipestone, where where you add, we know that that virus has the potential of moving from other countries to us here in the US. And once it lands in the US, through this transport network, can you then spread this virus across the US? And so I worked in collaboration. So we set up a collaboration with Scott D's group in Pipestone, and we simulated this this type of movement of feed across the US. And at various points in time, this transport truck stopped, and so it took 23 days to get through, and so it was experienced all these different conditions. The take-home message for us was that this virus was stable, and this was actually predicted under sort of theoretical parameters that it could survive this process. But for the first time, we actually had real data on a giant virus like ASF that showed that this virus is stable. But not only stable, we saw no degradation of the virus. The virus, we could get up nearly 100% back from what we started. And this is something you just cannot do if we didn't have this virus. And the surrogate system. What is also interesting about us applying this is that a lot of companies, as I said, originally got interested because they want to kill this virus, right? We know that temperature is probably not good enough on its own. We need to do a bit more than that. And so I was quite happy to enter in collaboration with other companies who have mitigants, who have some data on the other viruses that infect swine, like PRRS or PDV, but they had no data on ASF. And so we said, well, let's use the RISN assay. Let's use this algal virus. And I named 3 companies here: Probiotech, Walco, Chemin, and Parasol. They've given us samples to test. We have a few more companies that are lined up to do more of these tests. And this is sort of data we can produce, right? We can start again with like 1 million viruses, and we add their mitigant, and we ask, does it work? Some mitigants don't work, others work really, really well. And so and you can do time and dose experiments. So this is a plot here example of different doses, different ranges one uses. Some of the data here is in the presence of feed or not in the presence of feed. So all these dynamics we can replicate, and we can give real data as to whether some mitigants are effective and others are not. So this is the exciting time we're in now, right? We have a tool now that allows us to measure in a safe way how ASF behaves. We never had this before, and we're not limited by time. We're not limited by this select agent restrictions, and we can do this in real scenarios.
Fascinating. So if you have any questions, those folks who are joining us online, for Jerry or for Declan, please post those now. So Declan, what's the next steps? I mean, where do you go from here? You mentioned there's some more work to be done, and some more things to be learned. What are you looking at doing next?
Right, Sarah. So the idea is really trying to use this assay in as many scenarios as possible. So some studies we're trying to get off the ground is like looking at depopulation scenarios, right? So for example, if ASF had to come here, God forbid it doesn't happen, hopefully it never does. Mm-hmm. But if it does, we need to be prepared. And we have a strategy here in the US of saying, right, if an outbreak happens, how do we contain it? Can we clean that facility? And in doing so, are we sure not to exacerbate the problem? Are we sure that what we're doing won't create more virus spread? So if we had an example, say like temperature was good enough, well, that would have been a big mistake. But now we have temperature and we have mitigation strategies, these chemical mitigants. So what we want to do is we want to go and run depopulation scenarios, find out whether the equipment post cleanup can be cleaned properly. So when we move it to the next facility, it doesn't spread the virus. Transport, again, the actual equipment we use, what's going on there? Are our methods good enough for ASF? Then you have the scenario where we have this. So that's like the practical side of it, right? I'm giving you just one example here. Another question we really have is this interaction, especially between the virus and feed. We know that they bind to each other, and know they interact with each other. And we are trying to understand that relationship. And so the tools we're developing, so we're not resting. I like developing new tools. So we've got the viability assay, we've got this confocal assay, but there are other assays we can use in my toolkit as a molecular biologist to ask, how is this virus interacting, and getting to the mechanism, understanding how this virus interacts with feed. So for some cases, for example, a virus being present in feed might not be a contagious virus. It might be stable, it might be viable, but it might not be contagious. It might not be able to be spread because it's bound to a matrix, right? All these questions we can really start addressing, and we can address it using this RISNA assay.
Really comes down to being able to understand, as you say, that virus and how it interacts, and you know what it means when it's in feed, if it's viable or where we're at. So Jerry, what about key takeaways here for the feed and pork industries? What do they need to really pay attention to now that we have this new assay and new ways of measuring these things?
Well, again, I'm going to come back to one of my favorite phrases, and that is, I think our work is demonstrating that measures do matter, and the way that we go about measuring things, the choice of assay that we use, the interpretation of the data, I think I want to emphasize it. They're critically important because lots of very, very important biosecurity management decisions and protocol decisions are being made based on what we thought we knew in the beginning, but now we're realizing that time and temperature treatments probably aren't quite as good as what we once thought, and we're going to have to go to higher temperatures, and that becomes an issue because when you think about the nutritional value of feed ingredients, let's pick vitamin trace mineral premixes, for example. Anytime we start elevating temperatures, we start destroying the potency of some of our vitamins. And that goes contrary to the whole goal of what we're all about trying to accomplish in the first place. Some of our high crude fat types of ingredients or high lipid content ingredients are susceptible to lipid oxidation at high temperatures that are necessary to inactivate this virus. So that's a concern. So, I think what we're looking at going forward is some optimization of, yes, let's use a certain amount of time and temperature to potentially inactivate this virus, but we're going to need additional strategies such as chemical mitigants and so on to really be able to have the amount of confidence and control that we need to deal with any sort of contamination that might occur.
Very interesting. I want to thank both of you for joining us here today. It's been a great insight and a look at what's new out there when it comes to African swine fever virus and how we can detect and how we can better understand it. Always great to have you with us.
Thank you, sir.
Thank you, sir.
Mark your calendars tomorrow right here on 365. This will be interesting. We have a conversation with a company that is developing a new technology to make soybean meal more digestible and nutritious for livestock without having to rely on the traditional processing methods. So some of that could play into some of these things we've talked about here today. As always, all Feedstuffs 365 sessions are available in on-demand format on feedstuffs365.com. So if you miss them or you want to go back and rewatch or share them with your colleagues, feel free to do so at any time. Until next time, for Feedstuffs 365, I'm Sarah Muirhead.